Hi there. Today we will learn about genetic engineering on Study with Swarnali. As the name suggests, it's genetics plus engineering makes genetic engineering. In simple words, it is the use of techniques to create any kind of modification or change in the genetic material, that is DNA or RNA, of an organism in vitro by human intervention. That is, creating any modification in the genotype which will change the phenotype of the organism. Paul Berg is regarded as the father of genetic engineering. He won Nobel Prize in 1980 for his work on genetic engineering. He introduced gene of SB40 virus into bacteria using lambda fudge in 1972 for the first time, creating genetic engineering. Don't forget to subscribe. As you can see here, a part of DNA is getting cutted and new DNA part is getting added. This is called recombinant DNA. Now, there are some techniques or tools of genetic engineering. First is rDNA technology. It is the addition of foreign DNA part to another DNA, making a new combination, that is recombinant DNA, like addition of desired gene segment on a plasmid DNA. Now, gene cloning. It is making identical copies of desired gene, like the rDNA when enters the bacterium and replicates itself along with the desired DNA. Now, gene transfer. In this, the gene is isolated and introduced only the desirable gene without introducing the undesirable part, which is not necessary. Now, we will look at the mechanism of genetic engineering. First, plasmid is isolated from a bacteria, circular DNA. Restriction enzyme cuts the plasmid DNA. Meanwhile, human insulin gene is being isolated from human DNA. Restriction enzyme cuts DNA into pieces. As you can see in the diagram, ligase joins the plasmid and insulin gene. rDNA are inserted in the bacteria. The bacteria grown in Petri plate containing nutrient and antibiotics. The bacteria with the desired gene grows in the medium, and then insulin is produced by the bacteria, which are collected, processed and used. This is the mechanism of complete genetic engineering, as you can see in the diagram clearly visible here. The insulin produced is then processed and collected for further use in human. Isn't it a fascinating thing? The rDNA can be inserted further to plant or animal cells for making a change in their phenotype by changing their genotype, like pest-resistant plant. The organisms made from genetic engineering are called GMO, genetically modified organism. You can see some products saying GMO free and like that. Now, vector. Vector is a carrier of foreign gene, a self-replicating DNA molecule. Example is plasmid or viral DNA. There are some prospects of genetic engineering, like vitamin A rich golden rice from daffodil incorporated in the rice, carotene gene. Then it can replace the defective gene by genetic engineering. Then person specific organ cells from stem cells can be made for transplantation. It's going on actually. This kind of works are still going on, and many more. Now, genetic engineering is a very vast subject and a fascinating one. We will learn more about genetic engineering related things in upcoming videos. Thank you.